Welcome back, dear students, to vectors. We all know vectors are sequences of DNA which are being used to carry the genes of interest, thereby forming recombinant DNA. And they are the vehicles which carry these genes of interest into a host cell. Now, there are different types of vectors which we use, like plasmid vectors, phage vectors, cosmid vectors, phagemid vectors, and eukaryotic vectors. You know what is a plasmid? A plasmid is an extra chromosomal genetic element and found in bacteria, and they can be used to transfer some amount of gene of interest into a new host. And a phage is a, what is a phage? It's a what? You have heard about the term bacteriophages. Same way, a phage is a virus which can be used as a vector. The terms cosmid and phagemid are new to you, and we will be dealing with that in the coming sessions. And those vectors which use a eukaryotic hosts for its propagation, they are referred to as a eukaryotic vectors. So let's go into the details of the vectors. The first one that we are talking today is about the plasmid cloning vectors. The plasmid cloning vectors are basically plasmids, and they are circular double-stranded DNA molecules that normally exist in the bacteria and in the nuclei of some eukaryotic cells. Plasmids, they can replicate independently in the host cell, and the size of the plasmid in different organisms varies from about a few kbs to nearly 100 kbs. Now, the size of the plasmid can vary, but the amount of gene of interest or the size of the gene of interest with which it can hold is about 10 kb. That is, if you have a plasmid, the maximum amount of the gene of interest or the appropriate amount of the size of the gene of interest which can be inserted into the plasmid is of the size of 10 kb. And plasmids, as we know, they have the origin of replication. They have various selectable markers such as antibiotic resistance genes and also various unique restriction sites. And we can see that after the culture growth, a cloned fragment can be recovered easily, and the cells are lysed and the DNA is isolated and can be purified. So a DNA fragment can be kept indefinitely if mixed with glycerol in a minus 70 degrees Celsius freezer also. Now, you might think all these characters fussy and vague unless and until you see a vector. So let me just show you a plasmid vector. You know what is a plasmid? It codes for the these are extra chromosomal genetic materials, and they code for various characters in the case of bacteria. Now it could be antibiotic resistance, or sometimes it could be specialized properties like degradation of some xenobiotic or something like that, that can be coded by the bacteria if it contains a plasmid. Sometimes heavy metal resistance. These are all conferred to the bacteria by plasmids. And you have also come across different types of naturally occurring plasmids called the TI plasmid. The TI plasmid which has been found in Agrobacterium tumefaciens, or it could be resistance inducing plasmids, like as we mentioned about the antibiotic resistance and all that. So that is all naturally occurring plasmids. Now when we need a vector for the genetic engineering, we need to synthesize a vector according to our own interest. And moreover, you can also synthesize it in your in a laboratory, it has to be synthesized based upon our own needs. Because as we told, a marker, if a vector has to be used in genetic engineering, it should have many restriction enzyme sites. It should have a selectable marker. It should have an origin of replication. And when you consider every restriction enzyme site, it should have only one site for each restriction enzyme. Now look into PBR322, which was a plasmid which was being made by Bolivar and Rodriguez and two researchers, and the name goes to it, PBR322. What is 322? It refers towards a number which distinguish the plasmid from the other plasmids which were being developed in the same laboratory. It was, now just look into this. Here, this is the origin of replication. That is the site from which this plasmid can be replicated. So if the plasmid has an origin of replication, it can replicate in a host cell independently. Now look at this portion. This is AMR, that is ampicillin resistance. So all those bacterial cells which have this particular plasmid will attain the property called ampicillin resistant. Then here there's another one, TET-R. TET-R is a tetracycline resistance. That is also a selectable marker. So these two, ampicillin resistance and tetracycline resistance, can be used as selectable markers to identify these plasmids. So in all those host cells in which this plasmid is being attained or is entered, you can see that they attain a character of ampicillin resistance as well as tetracycline resistance. Then you can look into the names over here: PST1, Echo R1, HIN3, BAM-H1, SAL1, AVA1, BAL1. Okay, so all these are all sites for restriction enzymes. And you should see there are many restriction enzyme sites on this particular plasmid, but every enzyme is only having a single restriction enzyme site. That is, if you go to look into Echo R1, there is only one site where Echo R1 will cut. And there is only one site on this plasmid where the HIN3 will cut. So these are they are meeting the properties of what? A essential or a good vector. So this is an example of a plasmid, and normally plasmids can carry a size of about 10 kb into it. That is, your gene of, you can cut this plasmid at different sites. Okay, two sites have to be cut, and you can remove a part of this particular plasmid, and instead of that you can put your gene of interest at that particular region. Now, there are different types of plasmids which are naturally occurring, like COL1, COLE1, RI plasmid, and they code for different types of characters, as I mentioned earlier: multiple drug resistance, enterotoxin protection, and all that. So, using artificial methods in the laboratory for recombinant DNA technology, we have prepared some vectors, and one among them is PBR322, as I have mentioned here. Now, PBR322, it has a size of about 4,363 base pairs, and it carries two sets of antibiotic resistance genes, as we mentioned earlier, and it has a great variety of restriction sites, and it has a very high copy number. What is a copy number? The total number of these plasmids which have been present in a cell is referred to as a copy number. That is, they can be maintained in a very high copy number in a cell by using some techniques. Now, what is that? I need to tell you something. When you considering a plasmid is something extra chromosomal. That is, the bacteria can survive in a media even if it doesn't have a plasmid. So always the bacteria will try to get rid of the plasmid when it is continuing its life cycle. But in the point of when we are doing genetic engineering, if we have put a gene of interest into your vector, it becomes essential for you to make multiple copies of this plasmid. That is, you need that the plasmid as well as the gene of interest which it carries is being transferred to every generation. So what you do is, after putting your recombinant into a media, you will also put some antibiotic which is being coded by the vector so that the plasmid itself will get produced in large copies. So PBR322 can be maintained in a high copy number by using some selectable markers when it grows, like using either ampicillin or tetracycline, any one of them. Not both, you can use any one of them to what to increase the copy number so that the plasmid is not being lost from one generation to the other. Normally, E. coli has around there are about 15 molecules that have been present in a transformed E. coli. That is, in the E. coli, once after it is transformed, plasmids of about 15 copy number could be present. And if you want a higher number of plasmids in E. coli, you can also give another antibiotic called chloramphenicol, which is a practical tip which I have included here. Uh, you can increase the number of copy numbers of plasmids in E. coli by growing them in chloramphenicol, because at the presence of chloramphenicol, the protein synthesis is being inhibited, and so the plasmid amplification can be increased to about 1000 to 3000 times. These are regarding the advantages of PBR322. It has a very small size, so it is easy for purification and manipulation, and it can be amplified up to 1000 and 3000 copies, which these are all things which we have mentioned here. But the disadvantage is that it has a high mobility, and the size of the genes which can be accommodated into it has been found to be very less. So these are the disadvantages into it. I guess you understood PBR 322. So it's a plasmid, and into this plasmid we can put our gene of interest. Now there are other cloning vectors like PBR 327. Then I'm just going so that you get familiarized with the names. They could be PUC vectors, PUC vectors. The PUC vector is derived from PBR322 itself. It is having different vectors are there. Then it also has a replication origin and ampicillin resistant. But in addition to that, they have put another gene called the LAC-Z gene. And LAC-Z gene is something which is helpful to identify the cells which contain the recombinant. And PUC vectors can function both as the cloning vectors as well as the expression vectors. This is an example of the PUC vector. It would have a LAC-Z gene incorporated into it, and these are the restriction endonuclease site. That is, there's a large number of compared to PBR322, there are lots of restriction enzyme sites. There is a origin of replication over here, and there is another gene called the LAC-Z gene, which we will be explaining in the coming sessions. And there is also a polylinker cloning site also here. And there are different versions of PUC itself depending upon the sequence variations. Uh, there are different versions of PUC. Which version you want to take for your work is different. Now, coming to PUC, as we look here, PUC puck is a vector, as I told you, which is having a PB, just like PBR 322, it is having a sequence called the polylinker, which can recognize several restriction enzymes so that the vector can be used for cloning a variety of DNA molecules. And once you get a puck vector, okay, into the puck vector you can add a large amount of DNA samples, and if the puck is being puck vector is not containing, that is without containing a gene of interest, or if the puck vector as such goes into a host, it gets transformed into a host, it all those cells will attain a blue color. Now why it is, we will be explaining in blue white screening. And if your gene of interest enters into the vector and that recombinant DNA enters into a host cell, they will appear to be as white in color. Now, what is the reason for this? The reason for this, the presence of the LAC-Z gene. The LAC-Z gene, it codes for an enzyme called the beta-galactosidase. And when you grow the recombinant cells after cloning in a media after transformation, when you in the media, you use here two selectable markers. One, of course, ampicillin resistance gene, because the puck vector codes for that. And at the same time, you use another component called the X-gal. X-gal is a substrate for the beta-galactosidase. So, and if the insertion of DNA happens in the what in the laxy region, what happens, the beta galactosidase gene will become non-functional, and hence what happens, the colonies will fail to turn blue and they will appear to be white. But if a gene of interest is not being incorporated into the vector, the LAC-Z gene will be functional, and the what the substrate X-gal will be broken down by the enzyme beta galactosidase, and the colonies will appear to be blue in color. So after cloning a vector using a puck, after cloning into a puck vector and transforming that vector, recombinant vector, into a host, if the cells, now the host, now here the host cell which we use here is E. coli cell. This is a plate containing transformed as well as non-transformed E. coli cells. You can see that there are some colonies which have been found to be blue in color. The blue in color, the colonies which have been found in blue color, they are the ones which are referred to as what? The non-transformants. And the white colonies are those cells or those E. coli cells which contain the what? The recombinant DNA is being present in the white colonies. They have lost the lag-C portion, that is in the lag-C portion, some disruption has been happened, and as a result, what happens? They will lose their what? Beta galactosidase function, and hence the colonies will appear to be white in color. So this technique, which is utilizing the LAC-Z gene for the identification of recombinants, is referred to as the blue-white screening, and this is a blue-white screening depicted on a media with the antibiotic ampicillin. I hope you got a better idea of what is vector. We spoke about the plasmids, and we referred to plasmids PBR 322 as well as the puck vector over here. And in PBR 322, we identify cells based upon the ampicillin resistance alone, and in the puck vector, we also use another technique called the blue-white screening. So thank you for now. We will be continuing in the next sessions.